Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. So, today we will be starting the 5th lecture. So, just to have a little recap what we did in the last fragment of the 4th lecture. So, we talked about the references or the way you study a structure, a dynamic structure like human body, or as a matter of fact any animal system. What are the different planes, how you cut it. So, we talked about the 3 different planes: sagittal plane, transverse plane and the frontal plane. This is where we kind of wind up, and earlier to that we talked about the lateral part, proximal part, superior, inferior, distal end, proximal end, medial part, lateral part, and all those terminologies. So, when I wind up in the last class, I kind of felt that the frontal plane, I should have explained it much better for you people. So, when you talk about the sagittal plane, I told you that you are giving the cut like this, okay. So, half of the nose in one side, half of the nose in another side, half of the mouth is another side, half of the mouth in another side. Then when you talk about the transverse planes, you are taking the hacksaw, you are cutting the body like this, like this. So, you consider a body as a cylinder and you are cutting the cylinder like this. Now, talking about the frontal plane, where I felt that you need a little bit of a more understanding, is that when you are making the cut like this. So, suppose you give a cut like this. So, in the first cut you will have the nose, eyes and everything. The next cut you will have the part of the brain, and as you go back you will have the cut of the rear. So, that is what we call as the frontal cut. So, these are the different ways by which a 3-dimensional structure is being studied. So, every section what you get, you study the anatomy, histology of that section, and then likewise you add all of them and then you figure out the 3-dimensional structure. So, there is a lot of image processing which goes on in modern days anatomy, because this is exceptionally essential. Say for example, as I was telling you in the last class, on a 3-dimensional object, say for example a cylinder, somewhere in a space at a particular x, y, z coordinate, there is some ailment. So, how you locate it? You needed a reference point. So, you really have to image a person section by section. So, having said this, these are the olden days how anatomy has been studied, but the modern days anatomy is more on the side of imaging, non-invasive imaging techniques. How without damaging the system you can image the whole inside thing. It is almost something like an X-ray what we do. When you pass through the scanner, you are getting scanned by the X-ray, okay. Similar to that, but that only can do for the hard tissues, not for the soft tissues, okay. How we really can image a system without destroying it, and there is enormous amount of research which is going on all over the world for non-invasive imaging techniques, and this is one of the most challenging area, falls under multiple expertise, starting from optics to imaging to mathematics to image processing and to advanced camera and advanced microscopy. It is a very, very interdisciplinary area, but an area which will flourish in the decades or the centuries to come, because one of the things, just think of it, say for example in your body blood is flowing. So, it means it is just like you are taking a remote sensing image or something, from the top view you can see a river is moving. Exactly similar to that, within your body blood is moving. It is being pumped from the heart, it travels all over your body, comes back to the heart. So now, could you have an imaging scanner which can tell you at what speed blood is moving? So even without checking the pulse, without checking the blood pressure, just you can be able to visualize the flow of blood in a real system without doing any invasive application. So this is where modern days anatomy, which if you, those who are aware of Gray's Anatomy, these are very old books and almost biblical in terms of the way it has shaped the modern day medicine. But the modern day's anatomy is heading more on a different frontier, which is, of course, the basic anatomy hasn't changed, but much more life into it. It is no more a static subject. It is a very, very dynamic subject. And why not? Think of it. A cancerous tissue grows or a lump grows. It's a dynamic system. It is not a static system. At some point it is happening. Okay? So, those of you who have a flair to enjoy image processing or who have a flair of reconstructing 3D image from 2D slices, they may explore what all modern days medical anatomy or as such anatomy as such can offer. Okay? And there is another component to that, the depth. For any object there is a depth component. So you just do not have an x and y, it is a three-dimensional object. So there is a depth component and how you measure all these things. Okay. Having said this, today we will move on to the molecular organization, followed by the cellular organization and followed by the tissue organization of our body. So let's move on to the next slide. So in lecture 5, so today we will be dealing with, so let's put it, so we are into lecture 5 and the first thing today we will be dealing with is the chemical level of organization. So in these 3 sections what I will be dealing with, I will kind of brush up your basic, the chemical basis of organization of the body. Organization of the body. Now, if you look at it, we all are made up of atoms and molecules, and nature has picked up very simple atoms to design us. It has picked up carbon, it has picked up nitrogen, it has picked up hydrogen, it has picked up oxygen. Apart from it, it has picked up few other transition metals like iron, cobalt, manganese, molybdenum, but in a very small amount. And these different molecules or these different atoms eventually form molecules, and these molecules have self-assembled. Some of them are complex molecules, some of them are simple molecules. They have self-assembled to form the structural component of our body, and some of these molecules stand alone has formed the framework or the conditions or the base where all the reactions of our body takes place. Some of these simple molecules have taken part in synthesizing much more complex molecules. Okay. So just if we kind of, what I will be doing here, I will be outlining all those different molecules which are involved in the chemical basis of the organization of the body. So while outlining, so just now I talked to you about we are having series of atoms and molecules. And if you look at the different atoms which are involved, carbon, nitrogen, oxygen, hydrogen, iron, manganese, molybdenum, magnesium, chloride, sodium, potassium. There are a handful of them which dictate our system. Okay. Followed by that, most of these atoms are bonded with each other by three different kind of bonds. Bonding of atoms. Here I am just kind of brushing the basics, because eventually while we will be going through the detailed course, all these things will come very handy. So, you will have the ionic bonds, where full-fledged transfer of electron taking place. Then you are having the covalent bonds, and then you are having the hydrogen bonds. These are the key things which you have to revise, and I wish to request you, please pick up any biochemistry or go to any online, just read a couple of sentences about them, okay? Because we will be asking some small questions on these things, multiple choice of course, on these things. What are the ionic bonds, what are the covalent bonds, and I am not going to get in depth into it because these are being covered in several courses, but we will be needing all these things. So, just to kind of give you a refresher here. So, these are the points you should know, okay? So, from here we move on to what are the basic. So, all these different kind of molecules goes through a series of chemical reactions, okay? Rx stand for the reactions, and they could be either irreversible or these are reversible. And one more thing which I just forgot in the previous slide to mention you, these covalent bonds could be further classified into non-polar and polar covalent bond. So, among the example, polar covalent bond is like water, which is a polar molecule, and non-polar covalent bonds are the one which form the framework, most of the molecules which form the framework of our body. Okay. These are some of the points you should remember. And coming back to the next slide, you are having series of chemical reaction in the form of reversible, irreversible chemical reaction, and the roles of enzymes in this process. So, what I expect from you, you should, of course, we will be talking about a specific enzyme, as we will be moving like enzymes like carbonic anhydrase, nitrogenase, and series of them we will be talking as I will be moving through. But just have an idea, at least a very, very fundamental idea, what are enzymes, how the enzymes function, what is kcat value, and all those kind of things. I just wish you guys to kind of go through that as a part of your self assignment to brush all these basic concepts, okay? The roles of different kind of enzymes. Followed by that, we talk about the different kind of inorganic compounds which are involved in our system. And the major inorganic compound which plays a critical role is water, inorganic acids and bases like sodium hydroxide. And talking about water, all our reactions in the body, the medium is water, and that is why you call it aqueous system. All the reactions you can think of, all have the medium as water. Then we are having a series of salts starting from sodium salt, chloride salt, potassium salts, magnesium salt likewise. Then we are having series of buffer systems for pH control. These are very important, as we will talk about the different kind of buffer systems in the kidneys, in the lungs, in the blood, these different buffer systems. So, what I expect from you people, you should know the basic, very basic of what is pH, what is a buffer, and how these different buffer systems regulates the pH of the body. These basic knowledge I expect you guys to brush during this process. Okay, these are all you all have studied all these things in your school level. Okay, next from here we move on to, we talk about the organic compounds. Again, this is the basic biochemistry's first chapter, the different organic compounds which are involved, which includes your carbohydrates. So, I expect you to know what are the common carbohydrates. You have the lipids. We will talk a little bit more about the lipids, as we will talk about the bilipid membrane in the cellular organization. Then you have the proteins. Proteins could be classified at two levels. You could have structural proteins as well as you have the functional ones. Of course, both are functional, but these are the ones which are taking part in direct reactions, and these are the ones which are forming the structural components. Then you have the genetic machinery, which is run by the nucleic acid, and they have specific locations within the body. And then the most critical one, the energy compounds like ATP, ADP, NADPH, like ATP, NADPH, NADP, FAD, likewise. These are the difference. Of course, the most critical one or the most important one is the ATP. So, what I expect you people to kind of know the different kind of carbohydrates, especially why glucose is so significant. Lipids, in terms of the classification of lipids, you pick up any biochemistry text, you will be able to figure out nature of polar head groups and non-polar tails, the different kind of lipids which are present in the neurons, in the brain, ceramides. Go through any textbook, you will be able to figure that out. Then we will talk about the proteins, what is alpha helix, what is beta sheets. These are the conformation, alpha helix and beta sheet conformations. We talk about the different structural proteins, okay, like collagen, keratin. In terms of the functional, we will talk about the different kind of enzymes, except of course one of them, which is a nucleic acid enzyme. Okay. Then different other proteins which has different kind of functional roles. Then we will talk about the nucleic acid. What you people needed to know are the RNA and the DNA, and the energy compounds and where all they are involved. And when we talk about the energy compounds, we come in the very next slide about the different cell organelle where they are concentrated, all these different kind of molecules. Okay. From here we move on to cellular level of organization. Cellular level of organization. So, the biggest triumph of life form which has evolved on the floor of earth or wherever in the universe is the self-assembling of certain unique molecules to form a confined structure, what we call as cell. So say for example, suppose I just represent like these are the these different colors. What I am going to are assume these are the different kind of atoms which are there in the prebiotic soup. Okay, all different kinds of atoms which are present there, and this is the prebiotic soup billions of years ago. Out here at one point, by some physical forces of nature or something, somewhere or other, there is a self assembly which occurred. These could be complex molecules, these could be simple atoms, these could be molecules of lipids or something, and they form the first confined structure, what we today call modern day as a cell. And the cell is formed by the self assembly of lipid bilayer. Just few minutes back I was telling you I want you guys to know about the polar head group of the lipids and the hydrophobic tails. So here is the hashed part is the, just showing in the red, this is the polar head group which can interact with water, whereas the tail which I am showing in blue is the hydrophobic part. So the biggest triumph was or is how they self-assemble to form a structure, and on such a structure they develop systems of continuous different kind of transport mechanism, energy dependent transport, unidirectional transport, bidirectional transport, channels. They form a series of channels here, sodium channel, potassium channels, chloride channel, water channels, okay, and all these channels are of course made up of proteins, as well as they have marker molecules on top of these channels, help them to identify which are of carbohydrates. So if you see a membrane itself, so this is proteins which are forming channels, gates, pores, pumps. So what you see just in the previous slide, if you go back, so all these different molecules, what you see, there is one thumb rule which has governed all of them. They all have self-assembled somewhere or other, and we do not know how these different lipids molecules, or as a matter of fact the proteins, how they self-assembled, what made them the formation of the peptide bond, what were their situations, how such long chains were formed, how the DNA evolved. We do not know. We do not have an answer to these questions in the prebiotic soup, how these molecules were formed. We just have no idea, because this is far and beyond the time where we start saying that DNA, but how DNA evolved. Because if you look at the cell, in the left side I just give you this, and let me draw a classic cell. When you talk about a classic cell, if it is a mammalian, it's mostly a spherical shape. Okay, so this is the membrane, what just now, which is a bilipid layer, and you have a nucleus inside. Within the nucleus you have the nucleic acid Na, and this is the nucleus, and this is the lipid bilayer membrane. L stands for lipid bilayer, bi, and L stands for the layer. Lipid bilayer membrane. Then you have another organelle called mitochondria, which is taking part in all the energy production machinery of the cells. If it is a plant cell, of course, you will have chloroplast, which is harvesting the sunlight. So it's a very complex structure what we talk about. As a cell, it's a very, very complex structure, but how this complex structure evolved is one heck of a tough question. We really do not know how it has evolved. But what we know are these. Though we do not know the origin of it, but what we know is it forms a membrane, and this membrane has a semi-permeability. It does not allow everything to go in or go out. It has a lot of transport rules and regulation, as I was telling you. It could have a diffusion, it could have facilitated transport, and this part, what you see here, this is called the cytoplasm. Between the nucleus and this is cytoplasm, and these are the different organelles. Again, I am not going to in depth of it because I want you guys to brush your basics on this. And here you are having the nucleus, which is regulating your development and heredity. All these features, all the different aspect of a unique cell type are being conserved in this DNA. Okay? And through the expression period, each cell changes its forms and then finally rests at a final form before it dies out. And there are certain cells in our body which remain intact throughout our life, which includes nerve cells, cardiac cells, likewise. Okay? And if this is the cellular level of organization, the last and the final level of organization, what I wish to highlight here is these cells form different kind of tissues, which is called the tissue level of organization. And at the tissue level of organization, what we are having, there are 4 different kind of tissues we deal with mostly, which is epithelial, which is forming the cover of all the coverings. Say for example, all this surface, these are all epithelial cells of our body. Okay, one. The next one is connective tissue. We'll talk a lot about these connective tissues as well as epithelial cells in the sections to come. Connective tissues are different kind. They could be something called a proper connective tissues. They form a structural framework of our body. Then we have fluid connective tissue, which includes the blood and the lymph. Then we have supporting connective tissue, which includes your bones and cartilage. And in the very next section, post integumentary system, we'll talk about the bones and the cartilage. Okay. Third important tissue classification is the muscle tissue. Muscle tissue could be classified into 3 groups, and we'll separately deal with them, which includes skeletal muscle, cardiac muscle. Skeletal, I do not need to tell, all the muscles which are present in the body are the skeletal muscle. The cardiac muscle, the one which is handling your heart, and then you have the smooth muscle, which is lining the gut of the body. Okay. And the fourth and final non-regenerating one is called the neural tissue. So we'll have individual sections in each one of these things, and of course later we'll be dealing with tissue injuries and aging, tissue injuries and aging is the process where things differentiates. So what I wanted to highlight here while closing in on this class is that this is the framework where from molecular self-assembly at some point during our evolution cells were formed. The cells learnt a machinery to divide and replicate and conserves its information in the form of nucleic acids, and at some situation even in the form of proteins they could conserve informations, and they further develop into very complex structures of tissues. These tissues join together to form organs, these organs join together to form systems, and different systems form together or join together to form what we call as a body. So with this brief introduction of 5 classes of different level of organization, we will be hitting the subject from the mostly from the tissue level of organization, or rather the organ level of organization. So our next class will be on the integumentary system, followed by the bones and the cartilage. Okay, thank you.